dispersal. Exciting. All right, let me share my screen. All right, everybody see that?
Yep.
Cool. All right, so the last thing that'll be kind of covered on this exam is this notion of fungal distributions and dispersal, how they're getting around, and why we might actually care about that kind of stuff. So pictured here is from a pretty recent review paper. Kabir Pei and his team put together, looking at why the idea of how fungi are around is more than just an academic pursuit. It's not just scientists arguing with each other for no reason. Okay, so it's basically showing us what we can learn about the environment if we can better understand how fungi are distributed in the environment. So how biodiversity is affected. I mean, consider all the pathogens in the kingdom of fungi, all that kind of good stuff, decomposing effects. So we'll look at some competing ideas about fungal dispersal, and then why we might care. All right, so to start out, we got, these are kind of the three main competing ideas out there in fungal ecology. The first one is famously known as the Bos-Becking hypothesis, okay? And it's everything is everywhere, but the environment selects. Okay. Then we've got moderate endemism. That's another hypothesis, whereas they're saying there are a lot of cosmopolitan everywhere, found everywhere fungi, but a lot of them have restricted geographies. Okay. And then the strong biogeography, spelled that wrong, hypothesis, saying that most fungi reflect the same sort of population distributions as like animals or plants. They have the same major dispersal barriers, right? They can't get across oceans or mountains. Okay. So who's got thoughts on these concepts?
I'm going to go with the second one, because with us finding spores in the atmosphere with all those air currents, they can pretty much go worldwide, but that would still say that plenty have restricted geographies, because not all of them are going to spread like that or have spores that survive that long.
Okay. Who else? Any counterpoints to that? I guess my question then would be, what is of them?
That last one makes a good point about the dispersal barriers, because I feel like fungi can survive in so many different kinds of environments. It's about being able to get to that environment.
Okay. What would limit fungi from getting to some remote environment?
Well, just like it says, like oceans, mountains, like if they're relying on wind dispersal and stuff, they're not going to be able to cross the entire ocean with that.
Okay. Anyone else have thoughts on this?
Or it could be like the fungi that I found that it digests itself. That's just kind of laying down everything right at its feet. It's not wind dispersed, and so it's not going to travel very far either.
Yeah, it's not wind dispersed. I will point out that it attracts flies, though.
Well then, flies can get on boats, and boats can cross continents.
They can.
But how long does a fly usually live? Like, Would they
5
make
days
the
or
entire
something. I
journey?
don't know. But they can disperse the spores onto a bag of feed that's being transported to New Zealand, and then that winds up in a farmer's land on some soil, and maybe that was good enough if the spores lasted that long. This is kind of what drove the idea of everything is everywhere but the environment selects. It wasn't for first developed, it was for just any microorganism. The idea being that, like, if you're small enough, or your like propagules are small enough, they're gonna be getting around in the wind, or transported by birds or whatever. So it's not like, in elephants, they definitely make it to an ocean and they're kind of stuck. You can't accidentally track an Indian elephant on your boot into New Zealand, the same way you can with a microbe. So that was this idea, and it's gotten some fair bit of pushback. Boss-Becking has, because as we've kind of moved beyond morphological identification of protists and fungi, into molecular ID, we start noticing that there does appear to be, at least in some species, some restricted geography. Like, there's population structure genetically. It doesn't seem like there's a lot of mixing globally for some groups. You may have the same species of fungus in Asia and Africa, but they're not sharing their genes. They've been evolving separately. So maybe there are some dispersal barriers, at least for some fungi. So here are distribution maps of 2 microorganisms. Which of the 3 best explains each of these maps? So let's start with the top one here, Alotis podophyllii. You think everything is everywhere but the environment selects, moderate endemism, or strong biogeography when you see that map?
The last one.
Do you think strong biogeography?
Yeah, at least the coast waters and I don't know what other inland.
Yeah, and it doesn't make it down into Florida. There's like a hard cutoff there.
Mm-hmm.
Anyone else?
I think I'd have to agree. Just looking at
Okay.
where it's isolated at, I mean, they're not up against like the Rocky Mountains or anything
Right.
like that. So wind could easily take it across the rest of the United States, but it's not. It's isolated in those regions. So
Yeah, I mean,
I
wind
think
is
number 3 would be the best.
crossing the Great Plains there, so it does seem to have some strong, a, a definite cutoff for its geographic range. All right. So you're saying wind can't explain this?
You can't explain it with wind because wind can still travel farther west on there.
Yeah.
Unless you take into consideration maybe heat, but that doesn't make sense either because you're going to have the same climate, across. You'll have same climates in different areas even to the west of that. So,
Right.
I don't know. I'd have to go with number 3.
Okay. What about the second picture, the Metastelium gracile?
That one would certainly seem to support that second hypothesis. Everything is everywhere, but the environment selects. You've got that as diverse as Florida and Africa, New Zealand, Australia. That's a pretty diverse set of coordinates on that map. But there does seem to be certain patterns. It does seem to be in certain coastal areas. Actually, yeah, a lot of it's really close to a coast.
But we've also got like central Russia, the central United States. This is one that I actually worked on in grad school. It's a slime mold, and I can tell you right now that each of these dots are the places we looked for it. In other words, everywhere there was a sampling trip, we found it. So I would
So
bet.
learners don't trust maps.
What's that?
So don't trust maps. They don't tell you all of the information.
Well, yeah, they don't really. Since this, I found it in Alaskan samples also. So, like, basically everywhere we look, we find Nematostelium grassily. So to me, that's like a everything is everywhere kind of deal for this species. Its spores are real tiny, and they have like static electricity built up on them, so they can stick to bird feathers really well. So bingo, that can get around. What might be restricting, if it's not wind, what might be restricting a lotus up there in the top?
Seems to have low tolerance for freezing temperatures, or at least areas that have the varieties of biodiversity that thrives in really cold temperatures.
Mm-hmm. You're onto something with biodiversity there. A lotus is a rust fungus. So what's that tell you about it?
Its range is going to be limited by the species that it is endemic to.
Yeah, mayapple. Mayapple is the species that it needs, and that has the restricted range. If you were to overlay a map of mayapple distribution, it would be identical. So if you go out to the Rocky Mountains and collect some air samples, there's a even chance that you're going to find a lotus DNA. So the spores are actually getting out there. They're just not finding a suitable host. So to me, this still kind of sounds like the top one. Everything is everywhere, but the environment selects. Okay, and so what we're basically, with that hypothesis, we're saying that for the most part, at least given a long enough time period, these tiny spores are going to make it everywhere on Earth, but whether they actually flourish there is left up to conditions. So, you know, temperature, precipitation, the right host, all that kind of stuff. So here's some potential dispersal barriers for fungi. Just a lack of vectors. So if wind can't do it, if water can't do it for you, if the animals aren't there, okay, mountain ranges, oceans, deserts. And this interesting study from 2011, 95% of basidiospores fall within 1 meter of the cap. Pretty interesting study. They actually went to mushrooms and put out a bunch of slides on the ground, kind of in a little cross transect, different distances from it, and then put Vaseline on the slides so it would trap any spores that landed. Okay, so they did this with a bunch of different mushroom species and quantified the number of spores of that species across distance. And what they found was 95% of all the spores essentially are kind of just dumped right next to the mushroom. What does that tell us about mushroom dispersal?
It knows that, or I guess it's evolved that if it's successful in the environment where a fully formed mushroom can grow, then why not have more organisms in that area? Having it travel in a faraway place can't guarantee that a new individual is going to be successful.
All right, so you're suggesting then that it's like beneficial, like adaptive for Basidiomycetes to not travel too far from where they're doing a good job already.
Yeah.
Okay. Anyone else?
I kind of think that if it's all dumping within 1 meter of the cap, then it might be relying on on other things to pick it up and take it away, whether it be like we talked about, water or wind or animals. But if there's a lack of it, then I don't know, maybe it's hoping that it can get picked up by something that passes along. So
Okay. Let's think about the experimental design here. I don't want to disparage the authors. They did a good job, but, and this is a big but, they put down spore traps on the ground different distances away from these mushrooms. And they counted up the spores from that particular species right there, and noticed that 95% of the spores they counted were within 1 meter, and the slides that were further out had fewer on there. Okay, what spores were they actually catching? The ones that fell out of the air column. They weren't actually measuring all of the spores that were released from that mushroom, just the ones that landed. Does that make sense?
Yes.
Okay, so they may have been observing 0.5% of the spores, for all we know, that were released from that mushroom. Maybe it's more than that, maybe they got half, but if a mushroom is releasing 200 million spores in a day, some are getting around, right? So the way this experiment was designed actually didn't measure any of the spores that did get picked up by the wind. So it's a kind of a confirmation bias deal. But they did a good job showing that where spores were landing tended to be really close to the mushroom. Here's a couple other studies. Mushrooms use convectively created airflows to disperse their spores. They actually create their own wind. It's not like a giant gust that's going to turn your umbrella inside out, right? But it's enough to get spores moving. Okay, so by releasing these spores en masse, kind of all at once, it creates turbulence and basically a wind current. So that's getting them out of the kind of undergrowth up into the air column. Okay, we got another study here, ectomycota associated with arthropods from bat hibernacula. Okay, so they're finding fungi on insects in caves. How'd they get there?
I'm kind of going back to that theory where if about how can something cross an ocean, right? Well, if the spores are light enough in the air because they're so small, then why not have it cover great distances and even go into a cave or be transferred, or while it's in the air get caught up on an insect or a bird or a bat or whatever and actually fly into the cave and get released into there as well.
Yeah, that's what I'm thinking. What about this last journal article title down here, Not Poles Apart? Antarctic soil fungal communities show similarities to those of the Arctic.
Everything is everywhere.
Okay. This is where I don't know if you can tell, that's my bias, is tiny spores are making it around. If we're thinking on like, weekly or monthly time frames, no, right? There's going to be a lot of patchiness in distribution. But it takes about 70 years for our atmosphere to what's the word? Thoroughly mix. I couldn't remember the word mix. So it takes us about 70 years to have the atmosphere mix, and so in 70 years I would expect basically spores to have gotten everywhere. Okay. Another important vector for dispersal, humans. And so these scientists are computer scientists, not necessarily mycologists, but they use this as a way to kind of test out a new algorithm. So similar to like Google Maps and GPS wayfinding, they were kind of trying to do that and improve on it, and they used a road network in some islands off Sweden to show that the spread of a wild plant pathogen actually spread along the roads. There's really only one possible explanation for that, and that's animals using the roads, moving them, and that would be humans. Okay. So when you're really tiny, when your propagules are really tiny, the dispersal barriers we tend to make as much sense as barriers, especially if you look at it from a decadal time point. Okay. Humans move around, we take walks in the forest, we don't bleach our shoes before we get on an airplane, and then we step off in a new country, and boom, we've just moved some microorganisms around. Okay. So here's a really big meta-analysis, essentially looking at fungal communities, their distributions, and how different environmental, like large-scale environmental parameters can help explain the fungi that we've seen in these places. So explained variation by environmental variables. So this is comparing all these different communities to each other and saying, well, these two are this amount of different from each other, right? They share some species, but they don't share all these others, and they're found in different proportions. So it's kind of high dimensional math going on, but they're comparing entire communities to each other and then looking at how different the environments are. And so they can estimate how much is explained by the environment. All right, so we've got all these things here, like mean temperature of the driest quarter, right? Precipitation of the coldest quarter, gross primary product production, precipitation of the warmest quarter. So looking at really big scale variables like bulk density of the soil, pH. These are things that we basically like measure with satellites. That's how they got this data. And this is showing how much of it is explained, how much of the variance in these fungal communities. Again, I think this kind of goes back to everything is everywhere, but the environment selects. Okay. Closer look from that same paper, they're showing how important these different variables are. So like Bio 8 is pushing the communities towards this one direction. And Bio 8 is the mean temperature of the wettest quarter. Right? So that appears to have some role to play in what fungi are found where. Right? So this is again to say that the fungi are probably, at least most of them, fully capable of getting around. It's just when they land, can they survive? Is the environment selecting for or against? Now, different fungi have different traits, and we do see patchiness in distribution for some fungi, even saprotrophs that we would think would be pretty good at surviving anywhere. So maybe it's just trait dependent, right? So these different fungi have different sizes and shapes for their spores. Let's take a look at some of these. Here's Alternaria, shown relative to a pollen grain. So pretty small, but kind of a wacky shape. Here's Aspergillus. This is probably something that is growing on your bread when you were trying to get zygomycetes. Super tiny. Penicillium camemberti. That's how they make blue cheese. All right, real tiny relative to all these things. So some of these spores might be more amenable to traveling long distances, like Penicillium here, or some of these spores that are ornamented and have like little hooks on them that help them stick to animals. Pictured over here on the right is Gigaspora rosea. That is a fungal spore also. So really big, and it also develops underground. Arbuscular mycorrhizal fungi, those AMF, Glomeromycota, are famous for having really big, clunky-looking spores that aren't really ornamented, kind of develop underground. And so Dr. Chowdhury and her group did a pretty cool study very recently, just came out in May, basically asking the question, if these things have giant underground spores, how is it that they're getting around? Right? Because arbuscular mycorrhizal fungi are found everywhere that plants are found. How did they have this global distribution if their spores are big, don't stay airborne for very long, just kind of drop out of the air column? All right. So they did a pretty neat study using like green roofs with sterile soil and basically measured 20 meters up in the air column collecting these AMF spores. Okay, so the spores that could make it up the green roof were then found in these plants, right? Starting with sterile soil up there. So very cool experimental design. It took them a long time to pull this off because they had to wait for all these plants to grow and for spores to arrive and all that good stuff. Here's what they found. Essentially, the hypothesis was the ones that make it up to the green roof should be smaller, right? And they should probably be ornamented or even melanized. Why those two traits? The small one makes sense, but help me understand why ornamentation and melanization would be traits that we'd look for in air dispersed.
Melanization's for the UV rays. The closer you get to the sun, it needs to be more protected.
Yeah. Melanization is very important if you're going to be exposed to the sun at all, right? UV radiation causes thymine dimers in your chromosomes real fast. So melanization is protection. So the idea being, if you've adapted to aerial dispersal, we would presume that maybe there's some co-adaptation to being exposed to the sun, so maybe some melanization. And fungi are known to have melanin genes, same ones that humans have. What about ornamentation? No clue? Imagine the difference between a cocklebur seed and a strawberry seed. Which one do you have to pick off your socks, right? The idea behind ornamentation is to maybe help stick to insect vectors, that kind of thing, and so that's what they're looking for. So starting here at the bottom figure, this is the distribution in red here of all Glomeromycota as far as spore diameter, all right? So most of them, the mean is somewhere right in here, about 100 microns. We got some that are really huge, a few that are pretty small. Compared to the distribution of the Glomeromycota that made it onto the green roof. So what's that tell you? Being small does appear to be ones, microns. But for the most part, there was a difference between the fungi found on the green roof and the ground just below it. And that difference was the spores up there were from species that had smaller spores. Okay? They didn't find that they were more melanized or more ornamented, though. All right. So this brings me to my final little story. One of my recent papers came out last year where we actually went to the middle of the Pacific Ocean to Mauna Loa, so way up high, and NASA and NOAA have a bunch of experiments up there, and one of NOAA's experiments was this little PSAP machine, particle soot absorption photometer. And so they're essentially measuring the junk that's flying around in the air, the upper atmosphere, and seeing like how many particles per cubic centimeter, tracking that over time. Okay. One of the reasons to search for micrometeorites, which are constantly coming in. Every time you see a shooting star, it's exploding, and those little micron-sized pieces are actually floating around. Okay. So they're measuring that. So they're sucking air through a filter, trapping all these particles on the filter, and storing them in a sterile plastic bag. And so they had 13 years of these that they'd just been storing and were going to throw out. Okay, they were just kind of keeping them for no reason. And so we asked them if we could basically have these filters to look for spores. Right? And sure enough, we found a bunch of spores on these. So we did some microscopy work on it and saw spores. So then we just went straight to DNA, right? And we found a buttload of fungi floating around in the upper atmosphere, okay, above the inversion layer in the middle of the Pacific. Okay. We've got a bunch of plant pathogens. That was mostly what we were seeing, and then saprotrophs, and then kind of undefined things that could switch back and forth in between. All right. So here's a photo of a giant, ugly spore floating around that we pulled out of the atmosphere. How in the world did that get there? And so that was our question, essentially, is if we're finding big spores in the middle here, are they coming from Hawaii or are they coming from somewhere else? So on the days for those filters, we found essentially the old weather information, because this is a weather station. We were able to backtrack and use these little high split models. I forget what that stands for, but it tracks individual particles of air through time. Okay. And so we were able to basically separate out all the days where it could have come from Hawaii because of the airflow. And here's what we found. This is showing the last major landmass that air sample was over and how long ago it was. Some of them, 2% of them, we couldn't track back far enough to get to an open or a major landmass, but they're coming from Asia and North America. So what's this tell you?
Big spores can make it around too.
They're making it everywhere, even big spores. Okay, and this was a 13-year time series, and we saw stuff in every single sample. So over a decadal time period, bunch of spores. So we also wanted to look for like melanization, ornamentation, color, that kind of stuff, and we found nothing. Right? They were big spores, they were small spores, they were ornamented and not, they were melanized and not. It was just this kind of grab bag of fungal species in the upper atmosphere. Right? So the moral of these stories is that some fungi have a hard time getting around. The ones with really big spores, or that disperse their spores in water, right? Like chytrids, those zoospores, they rely on like a continuous path of water to survive and get around. But if you're making spores that enter the air column, you're pretty much good to go. They're making it everywhere. Whether they're surviving the journey is still an open question, right? If you're not melanized and you've been in the upper atmosphere for 6 months, you're probably dead. Okay. So where I'm coming down on the three hypotheses, and this is my opinion, is probably for the most part, all these spore-forming fungi that don't rely on water dispersal are getting around everywhere, and the patterns we see are because something about the environment is restricting which ones are surviving there. Okay. So that's your intro to fungal dispersal. Your assignment is to read the Bacigalupe paper. This one is a different kind of fungal dispersal because it is chytrids, and they don't make airborne spores. So look at that and how climate change is affecting them and amphibians. Okay. Then I will write up your Canvas quiz. That will be due next Tuesday. And then I'll post your exam to be due next Monday. Yeah. What is next Monday?
Will Tuesday be a discussion day?
Yeah.
What about Thursday?
Thursday I'll have a lesson. Next Monday. So yeah, the 26th for your exam. What's the date? Okay. And yeah, so next Thursday we'll be talking about endophytes. Any questions?
Thank you, Dr. Zahn.
All right, you guys have a good day. Good fall break and everything. And I'll see you next time.
Yeah, I hope you get to sleep soon.
Maybe next week.